Greetings, everyone, and welcome. Thank you all for joining us today for the HMSC Research Seminar. My name is Yi-Chung Chung. I'm the Academic Program Manager here at the Marine Science Center, and I'll be your host today for today's talk. Like all of our other seminars, this is a hybrid seminar event with folks attending both on-site and online. Roseanne and Rose are our hybrid support today, so please let them know if you're having any technical issues online so they can assist or let those of us on site know. For those online, we have your mics, camera, and screen share off. But please ask any questions you might have using the chat at any time, and we'll answer any questions at the end of this talk. If you put a question in chat, we'll read it out for everyone. You're also welcome to raise your digital hand at the end of today's talk, and we'll be letting you know when it's your turn, and we'll help to unmute your mic. For those in the room, if you have not already enjoyed a nice cookie and a cup of coffee, there will be some afterwards. Please make sure you sign in. There was a sign-in form at entrance. And if you haven't signed it, please sign in after. We'll be handling questions today at the end of the talk. If you have a question, please raise your hand. I'll bring this mic to you. And we'll have those questions addressed online so that folks online can hear. We are also recording this event, and all of our past seminars, as well as this one, will be up on our past seminar page, website, as well as on our YouTube page. Some just quick announcements. Next week's seminar on Thursday, December 15th at 3:30. We will have our Joe Haxel, a principal investigator with the Pacific Northwest National Lab. Joe will be talking about environmental monitoring of marine energy converters. Please join us in person here in the auditorium in the Gladys Valley Marine Studies Building or online for our last seminar for 2022. If you need info or login details for this or any of our events, you can log on to the HMSC homepage and scroll up to the bottom of the calendar and events with all the details. All right, so first, a bit about our speaker. Dr. Laurie Weitkamp has been a research fisheries biologist at the NOAA Northwest Fisheries Science Center since 1992. Laurie's work at the Northwest Fisheries Science Center primarily consists of salmon conservation, salmon research, and salmon management. She has been involved in salmon conservation through the Endangered Species Act listings of Pacific salmon, from the scientific basis for listings to periodic status updates. Her research focuses on the ecology of salmon in estuarine and marine environments, specifically how physical conditions influence biological processes that are important for salmon survival, including the likely impacts of climate change. A starting point for this work is understanding where salmon are during all stages of ocean residence, including the poorly understood winter period. Laurie served as one of the 2 U.S. members of the 2022 International Year of the Salmon Pan-Pacific Survey, which is a multi-year planning committee, and was chief scientist on leg 2 of the research vessel Shimada. And she received her bachelor's degree and master's degree and PhD degrees from the University of Washington. So please join me in welcoming Laurie to the stage.
Thank you, Chung. Can you guys hear me okay? Good. So as Yi-Chung said, I'm going to be talking about the 2022 IYS High Seas Salmon Survey. And I'd like to acknowledge my many co-authors. Ed Farley was my partner in crime. He was the other US representative to the Planning Commission. He was the other chief scientist on board the NOAA ship Shimada. And then the other people on this list were also part of the planning committee and or served as chief scientists on the other ships or were involved in other ways. And there's a whole another probably 100 people who are involved in this project in one way or another that I don't have here. So it's really a gigantic effort of this. So for those of you who aren't familiar with it and don't eat, sleep, and think about the Pan-Pacific Survey like I do, it is or was a well-publicized international multi-ship survey of high seas Pacific salmon habitats across the North Pacific Ocean that was conducted in late winter of 2022. And this figure is just showing the maps of the 5 ships that were out there conducting this survey. We had 2 from the US that are up in the northwest corner, the NOAA ship LM Shimada, and then the fishing vessel Northwest Explorer. We had the Russian ship Tinro, and then we had 2 Canadian ships. So the Sir John Franklin from the Canadian Coast Guard, which is their equivalent of the NOAA ships, and then the fishing vessel Ross Spirit. And collectively, these 5 ships sampled 131 stations across 2.5 million square kilometers of ocean. And I think that really, to me, says that it's really incredibly important that we have these international expeditions to do this. No single country is willing to spend that much money to do it. They could if they wanted to, but they don't. And it's by working together that we can do this kind of survey and really get that kind of coverage at once. And I also should emphasize that if you want to see a really good media machine, go to this website. So they had updates on Facebook, they had tweets. You go to the website, they've got all the information you want on the ships, they've got videos, they are making videos from it. In fact, there's a new one that they're working on right now that's showing what it is. It is really incredible. And I think as scientists, we're not very good at tooting our own horns and doing this kind of stuff. And this is a really good example. Camille Janaski was the one who was the main media person, and she really did a fantastic job. So it shows you what you can do. So what I'd like to do with today's talk is spend a little bit of time talking about why we did the survey. Why on earth would we go out there where nobody wants to go? Spend a little time on the methods, and then talk about the initial results, mainly from the trawling that we did, and then some of the preliminary analyses that are just starting to come out of it, and then end by looking forward. So as far as why we did the survey, I think a lot of you in this room know that there's juvenile salmon work that goes on in the coastal areas, like our JSO study, which is the map is shown here, where we're sampling juvenile salmon off the Washington and Oregon coast. And through these efforts, we have a pretty good sense of what goes on during this initial marine phase. We know where salmon are, what good ocean conditions, poor ocean conditions look like, et cetera. And this is an important period in their life. This is when we think most mortality is occurring, and we kind of have it dialed in. And there are similar efforts to the JSO study. The Californian Southwest Center used to do one associated with the San Francisco Bay area out in the Gulf of the Farallones. The Canadians do an extensive survey off Vancouver Island, northern British Columbia, and then the Alaskans, both the Alaska Fisheries Science Center and now ADF&G are working in Southwest, Central, and the Bering Sea, understanding juvenile salmon ocean ecology. However, they leave the coastal area and they move offshore. And in doing so, they enter what we call the black box, which is the high seas. And especially in winter, we really don't have a good sense of what is going on out there. And in fact, it is the least understood part of the salmon life cycle. So we don't understand where they are, especially on a stock-specific basis, or why they are where they are. We don't know who their potential prey or food, what they're eating when they're out there, who the competitors are, both salmon and non-salmonid predators. And then we know and would expect that there'd be pretty big north-south differences out on the high seas, but just because you've got this pretty good temperature gradient. But we really haven't been able to look east to west before, because normally surveys are much smaller. And so by having 5 ships out there, we can really look across a big chunk of the North Pacific and say, is it different in the east than the west, which is really cool. It's also been proposed that the high seas period, especially in winter, is a critical period because of expected low prey availability. So in the winter, there's not a lot of sunlight. In fact, there's very little sunlight. The water is just mixed up. It's a big washing machine. There's not, this shallow mix layer where prey is concentrated. And so the expectation is that there would be high mortality for salmon that basically don't enter the winter with a lot of energy reserves. So they run out of gas. And this would be especially important for fish in their first winter in the ocean because they're small. These are smaller guys. They just can't carry that much energy reserves and they don't make it. But this is really a hypothesis. This is kind of an armchair exercise. And the question is this really true? Is this what we're finding out there? And I think the bottom line is it's actually a lot more complicated than that. And then if mortality is high, what is the source of the mortality? Are they starving? Are they getting eaten? Or some combination or something else? We don't really know. So that's one of the big questions we've had. And then there's the issue of pink salmon. So pinks are the most abundant salmon in the North Pacific. This is the catch, commercial catch across all nations for the North Pacific. And you can see the pinks there probably make up as many salmon as all the other species combined. They're incredibly abundant. And they have this very strong even odd year difference. And admittedly, when you look across this, it's a little bit harder to see, but strong even odd year difference because they're completely independent life or year classes. The salmon, pink salmon grow to be 2 years old. So the odd years are completely independent of the even years. So you get this imbalance in many situations. Therefore, any odd year, even year difference people see in whatever they're studying, they blame on pink salmon, right? And so most of the work has come from sockeye salmon. Greg Ruggiero and Bev Agler at ADF&G have been doing a lot of this. This is just some figures on sockeye salmon production showing the different color bars are odd versus even. And they've made the point pretty clearly that there are big differences, even in odd. They've shown it with Chinook and coho and even killer whales and seabirds. And so if this is a winter high seas issue, which Ruggiero et al. Have said it is, then we would expect to see that pink salmon would be incredibly abundant and have high spatial overlap with other salmon. Again, is this really what's going on out there? And these kinds of studies don't just happen by chance. It really takes a lot of people pushing really, really hard, a lot of fundraising, making people want to go out and do this kind of stuff. And Dick Beamish has really been the one that has been pushing for this for a long time. If any of you know, he used to be with DFO. He's been retired for at least 10 years. And instead of going and playing golf, he's determined to go out to the high seas or get other people to go to the high seas in the winter for him. And he's really taken advantage of this long recognized need to go out there. He's got papers on it. It's also been part of the North Pacific Anadromous Fish Commission science plan for at least the last 2 cycles. And I know it's coming up on the 3rd. And so he organized winter surveys in the Gulf of Alaska in 2019 and 2020 pairing with Mark Saunders and Brian Riddle. And Mark and Brian are also retired DFO that are on doing other things. And these are just figures showing where the surveys in 19 and 20 occurred. This is salmon catches. I was fortunate enough to get invited to go out in 19, so I spent a month on a Russian ship, and that's how I got into this whole high seas thing. And there's also management questions that we need to answer or address with this work. So they don't spend this kind of money to go out and go look at myctophids, even though people who are into myctophids would love that. You have to have a link to management of commercially important species. So we've got that. So have changes to salmon winter ecology contributed to the long-term declines of some salmon populations, particularly those here in the Pacific Northwest? Is changing salmon or winter ecology responsible for these unexpectedly high and low returns that we've been seeing around the Pacific Rim? So the traditional methods of predicting how many salmon are going to come back seem to be falling apart at times, especially when we've gotten these marine heat waves. And one theory is that it's something on the high seas that's doing it. Can winter surveys help improve forecasts for salmon returns? This is the first step to salmon management, is knowing how many fish you think are going to come back. And having better information would always be good. And then from a strictly pragmatic view, which stocks would be impacted by IUU fishing. If you see a boat out there, what stocks are going to be there? We don't know right now. And so that kind of information is important for law enforcement. Okay, and I should say that we're not the only ones who have thought about going out in the winter out in the middle of the North Pacific. There's actually a pretty long history of that. The really nice paper by Kate Myers et al. That summarizes some of the data when they were out, what their findings were. And this is a figure that is from that paper that talks about all the factors affecting the winter distributions. And I think the bottom line here is it's complicated. But with that data, Skip McCannell, who's another retired guy who's still involved in salmon, has been putting together a data set from that historic data during the winter. So he's got the locations, the dates, the size of the salmon, some cases the temperature, a little bit of diet information that we can then compare to. On the left is a figure just showing who was out there when historically, and then on the right are the number of fishing stations that Skip has put together, just showing that there's this really big effort in the 60s and 70s, primarily by Canada and the United States. And then nothing happened during the 80s. In the 90s, there was some research, and then there really hasn't been anything since 2006 when the Japanese went out there, and they were just running the 145 line. So it's really limited, and then nothing until 2019 when we started going out again. And I think the real question is, we can use this historic data to ask the question, what is the same and what is different? That, you know, what should we expect? And is that really happening? If not, why not? So being out in the North Pacific in the winter is not the smartest thing to do. When I first went to go on the 19 cruise, I had no fewer than 4 people tell me I was absolutely insane. And this year, we really were insane. So this is the Winfield on the 3rd of September, or excuse me, the 3rd of February. And we were on the Shimada right off the coast. We were trying to get out to that black box, and you can see it was pretty rough out there. So this is, I think the red is like 80 knots or something like that. And when it blows that hard in the North Pacific, you get big waves. And in fact, we probably collectively as a fleet lost about 1/3 of our days because of weather. Either it's so rough you can't put gear in the water, or if you're trying to get from point A to point B, it just takes forever because you're bucking wind and waves out there. Another issue or another big challenge was COVID. I had nightmares for I don't know how long, thinking that, you know, we weren't going to be able to use the ship because too many of the crew are going to fail their COVID tests, and we wouldn't know that until the day before we sailed, when everybody had to get on. Luckily, that did not happen, but the policies were changing. And so in fact, between leg 1 and leg 2 on the Shimada, we had different policies on what we had to do to get on the ships and go out and sail. And then the one that nobody ever envisioned was global political events. So the Russians ended up pulling out. We were able to get everybody that was on that ship that didn't want to go back to Russia off. There were Canadians on the ship, I should say, and got all the samples off at sea. And so they could do that, but it is going to be a challenge, or continue to be a challenge with that. So what did we do out there? Across all 3 ships, we did 3 things in common. We did a full suite of physical oceanography. So we've got CTDs that went down. We're doing a full suite of whatever you do with physical oceanography, took a ton of water samples, et cetera. And I've highlighted environmental DNA there because we were taking eDNA from, I think, 5 different depths and a flow-through system. And the guy on the left there is Christoph Dieg, and that's what I call his milk machine. He would plug it directly into the Niskin bottles, and he could immediately filter a full Niskin bottle through 2 duplicate eDNA filters. And he had another machine that he ran through the flow-through system that I think he came from the Shimada alone. He had at least 300 eDNA samples. And it really proved itself because when it's so rough that you can't get a net in the water, you can get an eDNA sample. And so especially our leg 1, we had real bad weather, and we we had a lot of samples. And so it'd be really cool to see what shows up in those. We also had consistent biological oceanography samples. So we all did a standard vertical bongo net on all ships. And then the Shimada and the Franklin also did tucker trawls. And then the Russians did their Jude net. And we all fished. So everybody except the Raw Spirit used big ocean trawls. The Raw Spirit used a gill net because most of the historic sampling was with a gill net, and we didn't have a good conversion factor. And so what they did was basically follow the Franklin around and use the gill net where the Franklin was using their trawl so that we have a comparison. And from what I've heard, it looks like the catches are actually really similar. So both species composition and then the size of the fish were fairly similar because we assumed there would be some biases in there. Okay, and for those of you who don't know what these surface trawls look like, this is just from some drone footage they took on the Northwest Explorer on a really exceptional day. And you can see the surface trawls, so the buoys show up, and this is what it would look like underneath the water. So you've got the doors that spread to the net, and then the floats. And this is a really big net. This is a can trawl, which is bigger than the one that we use off the coast, the Nordic 264s. And you need a really big net because it's fish are few and far between. And we're all towing for an hour with that. And I love this photo. This is from, or a series of photos from Jackie King, just showing the different sizes of critters that we catch in the different sampling gear. So you can see for the bongos, you get a lot of copepods and krill and other weird stuff. The tucker trawls are a bit bigger, so we're starting to get shrimp and fish. And then the surface trawls, we get salmon, jellyfish, etc. In fact, I think we got collectively 2, maybe 3 salmon sharks caught out there with the trawls. So what samples did we take and what measurements? We did the basic biology stuff. Everything got identified to the lowest possible taxonomic group, counted, weighed. For the salmon, we took things like scales, otoliths, codaware tags, etc., checked gonads for maturation. And then we had a whole suite of samples for things like food web leakages and bioenergetics, stomach contents on a wide variety of species, and then duplicate samples of things like muscle, liver, etc., for both bioenergetics as well as using fatty acids and stable isotopes. And then we had some folks from the Southwest Fisheries Science Center out with us. They're really interested in thymidine because they've got issues with that with Central Valley Chinook. And so we took samples for that to look and see with a thymidine or thyminase, I can never remember which is which, levels are like. And then we used some of these newish technologies. So we did genetic stock identification on all the salmon. We took gill tissue for looking at the up and down regulation of genes and pathogens. This is some work by Christy Miller's lab at DFO in the NIMO. We took blood for insulin-like growth factor 1. What else we got? Stomachs to look for microplastics. And then we also took squid and mactophids specifically to look for microplastics. And this is going to be part of Olivia's master's thesis, which is really cool. And this is just what it looked like. This is some of the processing on the Shimada. I'm sure it was fairly similar on the other ships. Most of you probably know Marisa Litz up there, who came out with us. She's now with WDFW, but is still as good as ever in the field. Variety of samples, etc. So what did it look like? This is the temperature, satellite derived temperature and chlorophyll A for February. It looks pretty similar in March as well. And you can see the polygon in black is the sampling area. We had this huge range of temperatures. So it was about 3 degrees up when we were near Kodiak, all the way down to 9.5 degrees when we were at the southern end, and the current goes across the west wind drift, and then you've got the Alaska gyre counterclockwise. Chlorophyll everywhere we went was really low, which is what you'd expect in the winter on the high seas. So no surprises there. As far as fishing results, these are the results of the 22 successful trawls we had on the Shimada. And the point here is just showing you the diversity of things that we were catching. We never got just a species. So lots of jellyfish there, including a lot of the egg yolk jellies that look like big egg yolks on there. salmon, we got a lot of squid, some little guys. The very bottom right hand is our last set. We were down all the way down at 45 degrees, just a couple 100 miles off the Washington or Oregon coast, and we even got a sardine out there. He was probably as shocked as we were. So looking across all the trawls combined, this is just showing what we caught, and I kind of have them loosely grouped by taxonomic group. So we identified 55 separate taxonomic groups that kind of fall into these categories. We counted over 40,000 individual organisms, and that excludes jellyfish, because jellyfish get broken up in the trawls and you can never figure out how many you have. And that other invertebrate category is probably a bit inflated because apparently the Russians had one set when they got 15,000 salps. So I'm not sure if they really counted them all or not, but and then we caught almost 3,000 kilograms of content, so 3 tons, that mainly consisted of jellyfish and salmon. So interesting, pretty diverse groups of things. This is just the percent frequency of occurrence by either day in red or night in black for the different species that we saw. I don't have kind of the other invertebrate class on here because the taxonomy on the different ships was, they don't really always mesh very well. The Russians kind of use different names, et cetera. But you can see the diversity of things that we caught. And so the key here, these are just the percent of hulls on any of the ships that caught that species. So for sockeye and chum, somewhere between roughly 60% of the hulls during the day caught at least one chum or sockeye on them, et cetera. So you can see that sockeye and chum were both really abundant, frequently caught. We had kind of 4 species of jellyfish that were also quite frequently caught. So on the order of 50 to almost 80% of the hulls had at least one jellyfish in it. Lots of squid. These are all high seas squid, not things that you'd find on the up on the shelf. And then a variety of fishes with a lot of rare fish, including some myctophids, blue landronfish is the top line there. Some others. You'll notice that about halfway down there's or a quarter of the way down there's a dagger tooth, which is, as I will say in a minute, a predator on salmon, or known predator. But not a lot of ones of twos of things. And we also saw some pretty big differences between night and day, which you'd expect because we all know that things come up at night. One of the interesting ones was coho. So we got a lot more coho at night than we did during the day. And that's not really surprising. In 19 and 20, about 90% of the coho we caught were at night. So whether they're moving up or they can evade the net during the day, I don't really know. One of the jellyfish was also caught more commonly, the fried egg jellies, at night than during the day. And of course, you would expect most of the squid to be caught at night, as well as the myctophids. And then it was interesting that a lot of these kind of other weird fish that we just got a few of were also caught at night. And particularly in the case of salmon sharks, whether they saw the net during the day or they're up in the water column during the day, I don't know. But we mainly got them at night for the 2 that we did see. This is just a figure showing the distribution of the different salmon species across the study area. So the bubbles are the CPUE counts per hour towed, and the standard was to tow for an hour on all these nets. And then in parentheses is just the total number of that species that we caught overall. You can see chum and sockeye were the 2 most abundant species. It looks like sockeye are twice as abundant as chum, but there's a single circle that has a red circle around it. And that was the Russians caught, I think, almost 650 sockeye in a single tow. They're all little age 1s, but you can imagine how exciting that must have been to try and do something with that was meaningful. We did not catch very many pink salmon at all, 96, not even 100 pink salmon across the entire survey area. So that kind of blew one of our expectations out of the water. But you can see that they're really towards the southern end. They like the warmer water, as do coho salmon. We didn't get very many coho either. And I think part of that, and I think the Franklin was mainly fishing during the day. So they really didn't fish at night. So that probably has something to do with it. And then we caught very few Chinook, which is what we'd expect. Chinook are the least abundant of any of the salmon, and they tend to be deep. So they're probably Chinook underneath the net that we never caught at all. So if you take this data and plot it by haul, on the left, we have the Draftsman's plots. So each point is a haul, and it's just showing the relative abundance of all the salmon species. And then the numbers there are the correlation coefficients for that pair of species. And what's interesting is that pink salmon actually had the highest correlation coefficients on average with other salmon of any of the salmon. So it's kind of interesting. That's what we were expecting. We think they were going to be interacting, but there just weren't very many of them. And then if you take all the salmon data per haul and you put it in a MDS plot, and I'm just using the different ships here to indicate geography, basically, you can see there's complete overlap in MDS space. So the salmon are more or less equally distributed. I mean, there's some differences, but it wasn't like to the west versus the east. The catches were really different. They were all highly variable, but they were highly variable across the entire study area. Let's put it that way. One thing that was really interesting is that all the steelhead were caught by the gill netter. And in fact, it was their most abundant salmon species. None of the trawls caught steelhead. And I think we know that steelhead are really surface oriented. And I think what's happening is that when you're towing a trawl, there's a sag in the head rope, and it may be just a meter or two, and they're getting over it because they're really within that top meter or two. Whereas the gill net goes all the way to the surface. They can't get over the cork line. And so that explains the difference. And it really makes me realize, or wonder how many steelhead did we miss out there? And it would be really cool to get the eDNA and see, how many times were we sampling that there's steelhead eDNA. I should say that the initial eDNA, or not the initial eDNA, the initial steelhead genetics shows that at least 2 of those were Columbia River steelhead, possibly 3. So they're still working on it, but it's pretty cool. So we can also compare the salmon catches to what we saw in 19 and 20. And I have here the pie diagrams to show the catches, and I'm just using Shimada and Franklin because geographically it's the most similar to what we did in 19 and 20. You can see one of the really big differences is sockeye, that we caught a lot more sockeye in 2022 than we did in either 19 or 20. And if you know anything about sockeye, you know the big populations are the Fraser and Bristol Bay, and they're kind of on opposite trajectories. So 19 and 20 were the 2 lowest returns ever to the Fraser River. And the 2022 was the big year class for the Fraser. And then it's this off the chart Bristol Bay, 80 million sockeye returning to Bristol Bay in 2022. And so kind of explains why we probably got a lot. There's also temperature differences and a lot of other stuff going on. But I just kind of a quick look at what was going on. It's very interesting. As I mentioned, we caught a lot of other stuff. These are just distribution maps, same kind of deal. These up here, these are kilograms per hour showing the different species. And I think it's really important to think about, the entire ecosystem that is supporting salmon, because these are, in some cases, the prey, often the competitors with salmon. So we've got the 2 main species of squid, the 2 lanternfish, and then 4 species of jellyfish. And you can see that everything is pretty widely distributed. It's highly variable, but the variability is more or less consistent across the study area. There are north-south differences, but we didn't see big east-west differences, which is, I thought, really interesting. So if you take all the data from what I'm calling the main species, and there's a few others, sticklebacks, we actually caught quite a few of those out there. Some of the ships did. And you put it in MDS, and again, using the ship to indicate geography, you can see really complete overlap in MDS space for the counts on the left, which excludes the jellyfish, and by weight on the right, which really suggests that what we were seeing was more or less constant. It wasn't constant, but it was equally variable. Everything spread across the entire study area, which I thought was intriguing. The Russians have done a lot of work on predators on salmon out there, and this is kind of their laundry list of who they are. I pointed out daggertooths. I think we got collectively 5 daggertooths across the entire fleet, so not very many. Some sharks and marine mammals. And we really caught very few predators out there, either by nets, by eDNA, or seeing them in the case of the marine mammals. They just are not there, which begs the question, if predation isn't the source of mortality, then what is? And I don't have an answer for that at all. And I think it's really intriguing that maybe it's not, maybe there's not as much mortality as we think, or who knows what. You never see the bodies, as they say. But mentioning sharks, we did catch a shark on the Shimada. Sabrina Garcia was on board, and she's a shark expert, and she had tags with her. So I believe that's her with the yellow hard hat there that's facing the camera. She was drilling through the dorsal fin to put some satellite tags on. And it's always nice when you tag things that they stay alive. So this is the track. We called her Shakira. And you can see she went up to Prince William Sound and then came back and went all the way down to Southern California. And last I heard from her, she's still going strong. So pretty cool stuff. Okay, so what I'd like to do now is just really provide some hints of all the great things that are going to come out of this. We took a ton of samples. I estimated we took about 20,000 samples on the Shimada alone, of all the different tissues, et cetera, of all the different critters, and where people are just starting to run those and put the data sets together, and there's often divides, and one group is doing it one way, the other is doing it the other way. So we really don't have a comprehensive picture. But what we want to do is really trying to understand the entire ecosystem from the physical oceanography all the way up to the predators. And of course, the salmon, then we can drill in deeper and look at stock-specific differences or production type or age, whatever. And so I don't have complete data for any of these layers yet, but what I like to do is just give you some teasers of what it might look like. So this is some of the preliminary bongo or zooplankton results from the bongo toes that Alexei Pinchuk did. We don't have he did not have the Franklin, the Canadian samples. They're doing theirs, and then they're going to put them together. But it's just showing the distributions and really differences in abundances of some of the main zooplankton species out there. Assume these are all copepods. This is some of the preliminary genetic data from the Alaskans. So 2 genetic labs, ADF&G, and then DFO are running genetics. And they have different sets of markers because the markers we use down here don't work for Western and Russian populations. So the Alaskans are developing their own markers, and of course, they don't entirely mesh. And so this is the Alaska version of chum salmon. These are individual assignment results. Up in the top left is the showing the baselines. So the light blue are Japan, pink are Russian, dark blue are Western Alaska, orange are Central Alaska, Southeast Alaska, and then yellows are BC and Washington. And for each station, the pie diagram just shows you what the distribution of those groups were at that site. And I think it's really impressive how few of these circles only have one color in them. Often it's because there's only a single fish there. These chum salmon are totally mixed together. I mean, you can see, there tends to be more pink and blue, light blue to the left, more orange to the right. But we've got chum from Alaska and Japan and Russia all over the place. I mean, they're completely mixed together. And I think it's really, again, emphasizes the need to work together with different nations, because there's nowhere on the high seas that you can go and just get Canadian salmon or, Washington Northwest salmon. They don't care about international boundaries. And so it's really important, I think, that we pool our resources to try and understand what's going on out there. And it's really, really cool stuff. And I should say too, that granted, chum tend to be the most diverse of any of the species that we have out there. Another example of using genetic data, this is now from DFO. They're very interested in the 2022 sockeye return to the Fraser River, because this was supposed to be the big year class. So here is the different ships, the number of sockeye caught, and then they ran the genetics using their markers, the percent that are from the Fraser River, and then of those fish from the Fraser, what percent were from summer run versus late run. And the reason they're interested in this is that the late run usually contributes the most to the big year class. Historically, it's been 60%. You can see these numbers are really low, nowhere close to that. They had forecasts somewhere between 30 and 44%. And in fact, it ended up being 30% were late run fish. And so they were able to use this data to say, hey, think about what might be coming back, because it doesn't look like the late run is out there anywhere. This is a really, really expensive way to get this data, but I think it really shows you what you can do when you're looking across these really big areas and get this kind of data to get a sense of what is out there that will return. Digging in a little bit more to the salmon data, this is just some plots that I made showing length, fork length versus Fulton's condition factor. Fulton's condition factor is just a way to tell whether the fish is fat or skinny. So it's weight divided by length cubed. Generally, fish below 0.9 are really skinny, and you can see a bunch of these chum salmon were really skinny, really low condition factor levels. You can also see kind of the clear separation between the ocean age ones and the older fish for both chum and sockeye salmon, which have multiple age structures. And we need to use other methods to figure out how old the older fish are. It's interesting that both pinks and coho are only a single ocean age, but we actually saw a really large size range, given that they're more or less the same age out there. And I thought it was interesting. And then to dive into kind of these patterns, not just the size, but also the physiology, we have insulin-like growth factor 1. So Brian Beckman did not want to run these samples, but I said, I'll go get you blood. We'll run them. And this is some of the results that are just come out in the last month or so. This is sockeye IGF levels, and they're plotted by location. So we've got latitude versus longitude, and you can see that just the Shimada and the Franklin were the only two ships that took the blood samples for this. And you can see some geography in the pattern of high versus low. So kind of north, the IGF levels were high, down to the south, they were low. So it'd be interesting to dig in and see what's going on there. And then the sockeye, of course, is doing something very different than the chum salmon as far as IGF. So this is not location, this is the condition factor on the bottom versus IGF. And you can see that the Franklin and the Shimada samples are completely mixed up. So there's no strong geographic signal in this. And we've got some very low condition, very low IGF levels. You have some not great condition, relatively low IGF levels, and then some low condition, but high IGF levels. And Brian's most recent thinking on this is that it takes a while to get skinny, but as soon as fish eat something, their IGF level goes up. And so he's predicting, and it will be interesting to see if it's true, that these low condition factor but high IGF fish have stuff in their stomachs, whereas these ones down below probably don't and haven't been feeding. So it'll be really cool to see what comes out of this. And obviously stock is part of this, and he's starting to pull that apart. This is some work by Jillian. We mentioned the plastic looking in myctophid stomachs, and the upper picture is just us taking the samples. We got to wrap them in aluminum foil, etc. But some really cool pieces of plastic that have been found in the stomachs, which is kind of scary because we're way the heck out there, and to get plastics. Oh well. So this is just a really quick example of what we can do and what will happen with looking at the entire food web. This is some work from Jennifer Troina, who's a postdoc at UBC. I don't know how many of you saw her talk at the Monster Jam this fall. She's gone through and looked at 19 as kind of a starter, and she'll do the same kind of thing with 2022. But in 19, she found that there was a real break in the area with respect to physical properties, water column properties in the area we sampled. And that in the Northwest area, it was a very simple food web, krill dominated, therefore there's a lot of competition between the different fishes. And this is based on diets and stable isotopes, whereas out in the southeast part of the study area, it was a much more diverse food web, and therefore there was a lot less competition that different species were kind of able to stabilize. So I'm really excited to see what she comes up with for 2022, given that we've gotten this gigantic area, 2.5 million square kilometers of samples from. And then finally, I mentioned we've got this historic data. This is a really cool figure that Joe Langen put together. He's I think he's a PhD student at University of Alaska, and he is taking this historic data and playing around with it and coming up with models predicting where things would be. And this is his prediction. The shaded areas, the prediction for pink salmon catches. So the warm areas are where, based on the historic data, using the temperatures where they were catching them, we would expect to catch pink salmon. And then the bubbles and all the X's are where we actually caught pink salmon. And it's kind of interesting in that the areas that are purpley, there's a lot of X's up there. It's too cold for them. You wouldn't expect them there. But what is really interesting is this down in the bottom left-hand side, there should have been a lot of pink salmon there, and there were not. And what is going on with that? Why? What? I have no idea, but obviously something to dig into. So looking forward, we've got lots of samples still to run, data to analyze. We're going to need more meetings. I can't tell you how many meetings I go to for this, just checking in and et cetera. I see Olivia's smiling. You've only gone to one of 15 different types of meetings, and really trying to synthesize these data sets to make sense of it. And I think it's Dick Beamish who said, why stop now with these high seas expeditions? So he is actually organizing and fundraising to get a First Nations tribal staffed cruise in April on a chartered Canadian boat. And there's something called the Basin Event to Coastal Impacts, or BECI study, that they're working on, that they're talking about in 5 years from now, having another big Pan-Pacific. They've gotten approval through the UN Decade of Ocean Sciences, and it's going to be this ocean intelligence system where they're really trying to link not just the high seas, but the high seas to the coastal area, and do that. And with that, I will open it up for questions.
Wonderful talk. Questions in the audience?
Thanks, Laurie. It was awesome to hear the synthesis of all of that time at sea. I'm so intrigued by this, the fact that you didn't catch any pink salmon, but they remain like the highest in what's caught in the fishery. Is that right? So I know you talked about comparing to the historical pink salmon catches, but what are your musings on the fact that the fishery catches a lot and you caught very few? Do you think that has to do with where differences in where different boats are fishing or how they're fishing, or curious about that?
Yeah, you know, it's really puzzling. So Prince William Sound, I mean, is a huge pink salmon machine. Southeast Alaska is a huge pink salmon machine. The Russians, Kamchatka is a huge pink salmon. We should have seen pinks. And in 1920, we didn't get very many pinks either. And Vladimir was saying, oh, they're further south. You didn't go far enough south. Well, we went south and we didn't get any pinks this time. And other people said, oh, you should have gone farther west. We went west and we didn't get any pinks. So I don't know. I think we're all scratching our heads over that. And it will be really interesting to see what I have not seen I have seen the genetics, and I can't remember what they are. Unfortunately, genetics of pinks are not very good. And so there's Beringia, basically Western Alaska and Russia, odd year, and then everything else odd year, and then even year have differences. And that's the resolution that we can get with pink salmon right now. So the genetics is not great, but yeah, where are they? A lot of people are really stumped. We thought for sure, because we went further west this time, we went further south, we would have picked them up, and we did not see them. So I don't know. And you know, 2022, who knows what, I mean, the returns seem pretty good to the places that have lots of pinks. I don't know. I think we're all stumped. I don't think so. Yeah. You know, the Russian net that they used. Yeah. So the question was, is it how we're fishing? And the Russian net is designed for pinks. So it's actually pretty short trawl, and they didn't get any pinks either. I mean, they're the ones who are, you know, that where those X's are in the bottom left, that's where Tinro was. So I don't know. And because they're the ones who've dialed in the pink salmon catches, and they do more high seas work than anybody else. So I think we are all really puzzled what is going on with that. Where are they? Yeah.
Other questions online?
First of all, just a quick clarification. IYS, was that late winter 2021?
Yeah, so International Year of the Salmon is a even though it's a year, it should be years, plural. It was a multi-year organization or event that was global. So it involved the Pacific as well as the Atlantic, and they had similar, they didn't have a big cruise, but they had a lot of events associated with salmon, Atlantic salmon in the Atlantic, and it just ended this year. So they're now done with IYS.
Great. I have a quick follow-up too. So was there any acoustic sampling for marine mammals?
We did acoustics, just using the ship's acoustics. I guess if you have whale calls, you can probably hear them, and I'm totally out of my range with this. I don't know anything about acoustics, except that when we had the hydrophone on, you could hear the bing, bing. We were running acoustics all the time. So most of the ships were recording it. And actually Brandon Chasko, I don't know if he's on or not, he's working, and he and Brian Wells are working with the Shimada acoustics. And then there's a guy at, I think he's University of Victoria, who's doing the Canadian acoustics. So they're going to be looking at that. And I don't know, Don, can you pick up whale sounds with standard acoustics, ship acoustics?
I don't know if I'm the one to answer this question, but I think the acoustics that you're referring to that run all the time are active acoustics, active acoustic echo sounders that are pinging, and that's typically for mapping prey, and that is at very high, those units listen for the returns of very high frequency pings that typically don't pick up things like vocalizations of marine mammals, which you need passive acoustic monitoring. So it would be like either mounted or towed hydrophones. So the technology is a bit different.
Gotcha.
I don't know if that helps.
Yes, no, that does. Actually, that's great.
Thank
Yeah,
you.
I know we had there were times when there was a hydrophone on, and I'm not sure who was doing what with it. So the NOAA is a very complicated, or the Shimada is a very complicated ship, and they have lots and lots of electronics and like beyond my pay grade. Rick.
Yeah, Laurie, I was very interested in when you had the list of all the species you caught for the fish, the other fish. The third most abundant one was three-spine stickleback, which you had mentioned was, and I know they've been reported out there. They're actually caught them out in the old days of the gill netting, but it seems very bizarre that a fish that's like 2 inches long was able to get out in the middle of the high seas and actually, spends a lot of time in freshwater. So do you think that those are lost to the population, or?
No, so they were getting, we got just a few sticklebacks on the Shimada. Both the Franklin and the Northwest Explorer got like thousands in a haul. I mean, it wasn't a few lost, it was gigantic. And they got, I don't know how many sets. I mean, what's the frequency of occurrence? It's actually pretty high there for your sticklebacks. What is that, 15, 12%, something like that? Oh, it's actually more than 20%. Yeah, they were, yeah.
Yeah, but what?
I have no idea. I mean, there is,
areas?
it's interesting too, because Kate Myers had a student who was looking at high seas steelhead diets. And like the #1 thing was sticklebacks.
Yeah.
And it's like, what?
Yeah,
And sticklebacks, so the reason they're called sticklebacks is they have these spines that they stick out, right? They have dorsal spines that are really sharp. I've been stuck before with sticklebacks. They stick to your fingers. I mean, they pierce your skin. And then they have a spine on the side. So they're they're like eating blowfish, right?
Yep.
I mean, they would not be good, but yeah, no, they were
don't look
getting,
like they're very strong swimmers.
No,
How would they
no.
must have just gotten,
and there are big
Sure enough.
schools of them. Yeah, I mean, we got things like, black rockfish. You
Right.
see like 1 or 2 and it's clearly lost and got evicted off there. And
yeah, and I think, like you have the northern sea nettle, which is a very inshore species, and I know they caught a lot of those in the 2019 survey, but you would never expect those way out in the middle of the ocean. So it must have been a currents for decades.
we got those. I think though, where is it? If you look
Yeah,
at the figure of where northern sea nettles are, I mean,
Yeah.
we were getting them way out in the middle of nowhere.
Yeah. And they
So
can't-
from I should say that kind of from our southernmost point on that, was it the 147 line on the Shimada, we were only 200 miles farther from Hawaii than Newport to get back. I mean, we're way out there and we're getting sea nettles.
Yeah.
Yeah, so I think the idea that they're coastal maybe needs to get reevaluated because they're everywhere.
Yeah,
right?
they can't reproduce in deep water, so they're gonna
Yeah.
have to, those adults probably aren't gonna make it, I would think.
I
They
don't
have
know.
to- They have to
Yeah,
have some kind of
yeah,
substrate that they reproduce,
yeah, yeah,
so.
Yeah.
It's interesting.
yeah, yeah, no, that's a lot of lost sea nettles. Let's put it that way. But yeah, the sticklebacks are really puzzling and it wasn't, I mean, we got 1s or 2s, but they were getting whole nets full of sticklebacks. So,
Other questions? In the room, online?
So it was interesting that salmon sharks were primarily caught at night. Were there other notable species of sharks that were encountered during the data collection for the work?
nope, that was it. We didn't even, in 19, we got a couple dogfish, and I think at 20, there were also a couple dogfish caught. And we never saw any across the fleet in 2022. I mean, they weren't out there. So, and that's where the eDNA comes in. And Christoph hasn't run the eDNA yet. For 2022, it'd be really interesting to see what we pick up. But when he did do eDNA in 19 and 20, I mean, there were a few more salmon sharks detected and dogfish detected, but not many and nothing else. So yeah, it's really intriguing.
Yeah, we got more. Should you see whales through, I'm not going to do this right. Should you see whales though on an EK 60 or 80 with the right settings? I have no idea what that means.
No,
Okay. Thank you.
not unless it swims below the echosander.
Okay.
Yeah, I should say on the Shimada, I mean, we spend a lot of time looking out the windows. So anytime we're going to put gear in the water, we have to do marine mammal watches. And then as we're towing for our hour or two, and until the doors are pulled up off the nets, we are watching, and we've got like 5 or 6 people watching from the bridge at all times. So we had a lot of time, and we saw whales twice in 30 days of sampling. I mean, they just were not there. And yeah, it was impressive. And it was fairly similar when I was out in 19, a handful of whale sightings, and they're always too far away to see what they are, but you see the blow, and that's looking a lot. So we didn't, unfortunately, have a dedicated marine mammal observer on board. I really wanted to find somebody, but I couldn't because they were busy. But next time, yes.
No further questions online or in-house, so we will end our Q&A session. Thank you, Dr. Laurie Weitkamp, for this great presentation.
Thank you.